Evolution of Gene Expression in Animals by Khadija, Emma, Christie, and George P. Burdell. How do patterns of gene expression reveal the common evolutionary history of many animals? Here we have a phylogenetic tree of life, and in this presentation we'll be focusing on the animal branch. Animal classification. Animals can be split into two main groups, vertebrates and invertebrates. Invertebrates are animals without a backbone, such as mollusks, sponges, echinoderms, arthropods, and more. Vertebrates are animals with a backbone and are represented by chordates. If all animals evolved from a common ancestor, what homologous traits cause them to all develop so differently? How do patterns of gene expression reveal the common evolutionary history of many animals? Homology exists at many different and independent levels: gene structure, expression pattern, developmental mechanism, and anatomical structure. Homologous genes are genes from two different species related in sequence due to a common descent from an ancestral gene present in a shared ancestor. Hox genes are an example of a homologous gene in animals. They are a group of related genes responsible for controlling the development of an embryo's anterior-posterior axis, and they are present in all animal species. All animal body forms are regulated in similar ways by homologous clusters of Hox genes. The common ancestor of bilaterally symmetric animals, Urbilateria, is believed to have had 8 Hox genes based on gene sequencing of vertebrates, echinoderms, arthropods, nematodes, annelids, and flatworms. Throughout the animal kingdom, structurally similar Hox genes, orthologs, occupy similar positions within the clusters, showing that they are homologous. Through gene analysis, it can be seen that many phyla have gained extra Hox genes through duplication or lost them through deletion. As this figure shows, throughout evolutionary history, many different animal phyla have gained Hox gene clusters or lost them over time. Ed Lewis was the researcher who initially demonstrated that Hox genes exerted genetic control over segmental patterning in fly larvae. Lewis demonstrated that genes at the beginning of the clusters were expressed in the anterior regions of the embryo, and the ones at the end of the cluster were expressed in posterior regions. This suggests that Hox clusters arose through duplication and divergence from a common ancestor. More Hox genes or clusters typically correlates with more developed organisms. Compared to their common ancestor, protostomes have lost Hox genes, and deuterostomes have gained Hox genes and many more clusters. This figure demonstrates how protostome Hox gene expression has developed over evolutionary history. Protostomes typically have 3 to 5 Hox genes, as can be seen in the figure and the specific animals that are represented by the figure. Deuterostome Hox gene expression can be seen throughout evolutionary history in this figure. Typically, deuterostomes have 6 to 8 Hox genes, and many species have multiple clusters of the Hox gene. In comparison to protostomes, deuterostomes have many more clusters and Hox genes and are much more developed than protostomes typically are. Another example of the evolutionary implication of altering Hox genes was carried out in Drosophila by altering their UBX gene, which is responsible for hair patterns on the legs. By reducing or expanding the domain of the UBX gene, researchers could change the extent of hair on the fly's legs. In arthropods and vertebrates, Hox genes are expressed in derivations of all 3 germ layers: the ectoderm, mesoderm, and endoderm. In annelids, Hox gene expression is only detected in the nervous system. Thus, it is believed that the original site of the Hox gene expression and the last common ancestor of protostomes and deuterostomes would have been in the ectoderma, specifically neuroectoderm. Dr. Lowe was the researcher who demonstrated this by showing that the earliest sites of expression of Hox genes in Drosophila are in the ectoderm of the embryo and in the nervous systems of hemichordates and vertebrate embryos. These two pictures demonstrate how different expressions of the Hox gene can lead to different developmental patterns. On the left, there's a chick embryo where the Hox XC8 gene and Hox XC6 gene are displayed in yellow and red. Where the red and yellow overlap, there are ribs, but where the yellow extends out in front of the red, a forelimb develops. On the right, there's a snake embryo where there is no separation between the Hox XC8 gene and Hox XC6 gene. This leads to only the development of ribs in a snake and no forelimbs at all. A few takeaway points from this presentation are: 1. That Hox genes were present in the last common ancestor of the animal kingdom. 2. That they control the anterior posterior development of all animal embryos. And 3. That the duplication, alteration, and deletion of the initial Hox genes in developing animals are the driving forces behind evolution of the new species.